Right, there is a big typo. This is lecture number 5. So I'm just going to correct it. The reason the typo is I always copy the last slides and update it. The right information is here, lecture number 5. Okay, so today we are going to learn more about biology. We learn a little bit about chemistry. Um, just a little bit overview of biology. We're not going too deep, but at least it will give you some context how we are going to use biology for different environmental engineering applications. So today we learn about biology, and next class we'll learn the how biology can be used to understand toxicity. Because that's the toxicity assessment is big part of environmental engineering. If we think of now, how do you know what is more toxic, what chemical is more toxic, or another way to say is why certain virus is more toxic, more deadly than others. So these are the things that we need to learn more. And so to this week class is all about that. So that's the biological context. So this is week 3. After week 3, we are going to apply the same concept over and over. I forgot that now. This is the week also about a little bit background. Um, okay, so just a little bit announcement that homework 2 is due on Thursday. If you need more time, just let me know. Always I want you guys to submit. I do not want time should be a factor for your submission. So in case you have difficulties for any personal reasons or anything that happens, just always send me email. Do not just not submit, because that you lose point for not submissions. As long as you let me know ahead of time, I don't take any point out. So all you have to do is just ask. So today again, we are going to work on the biological aspect of it, and that's based on the reference book, because the textbook doesn't have those informations. So what we cover today is on chapter 3 of the reference book, which I already uploaded on CCLE. You should be able to have access to that. Right, so learning objectives. Now, the first thing you want to learn is what kind of different applications we'll have for biology in environmental engineering. In fact, when you think of environmental engineering, 90% of the work we do is either related to chemistry or biology. And then another, I would say not 90%, another 20 or 30% is physics or hydrology. So that's more on math side. So if you are good at math, if you are good at chemistry, or if you are good at biology or one of them, you have a very good chance in environmental engineering to make an impact. So you don't have to be good at everything. Just one of them is good enough. So today we are going to learn a little bit about biology. That's not my strength. I'm the first person to admit that I didn't like biology when I took the class in my undergrad. The reason is the way it was taught in undergrad in India is very different from how it is taught here. There we have to memorize every single um, the biological name. Now, for instance, you can't call frog as frog. You have to call them as the whatever that Latin name. I forgot what is that name. So it's a lot of memorizations the way that taught us, and I really don't like memorization ever. So that's why I had a perception that biology is really bad, because you got to memorize it. There is nothing about thinking or understanding. But I was wrong. Um, when I start learning about biology in my master's in environmental engineering, I begin to like it, because there is a lot of stories, because it's the story of us, how we evolve, how we work, how we functions, all that. So it's very important for you to understand those concepts. I'm not expecting you to remember a lot of stuff, but just understand how the food is consumed in our body, how energy is produced, where it is stored, what affects us. The same thing, the process is very similar for also smaller microorganisms like bacteria, fungi. It's more complex in our body, but it's much simpler there. So you'll understand those. So the second concept is you got to understand what different types of biological organism we are dealing with. That's section 3.7, 3.10, and 10 in the reference book. There we talk a little bit about just the overview of bacteria, fungi, protozoa, viruses. Again, I'm not expecting you to remember all the details in the textbook. Just know the brief knowledge, so that you know that these are the things will be useful when you apply those in future chapters. Most importantly, you have to remember why it is important to know those microorganisms, because you are going to either use them for a benefit for us, for instance, remediations, or you might be using them as a target, pollutants, to remove them, to kill them. For instance, coronavirus right now. How do you kill coronavirus? If you don't understand what's the virus made of, how it works, it's very difficult to understand the defense mechanism or how you can kill them. So then again, biology at the fundamental level is chemistry. So we'll understand the what's the chemical composition of the small part of different part of the cell or biological organism, and then we'll apply those concepts for energy and metabolism. For instance, how we consume food. Here in environmental engineering concept, food is actually pollutant, because you think a lot of pollutants are there which are organic. Lots of microorganisms actually can eat them for energy as well as their growth. So we'll understand those concepts or understand the math behind it, so that you can calculate how much contaminants can be degraded at certain conditions. Mathematics of growth, that's something like, it's important for many system where you have to grow the microorganism for them to work for you. So you got to understand the math behind it, and that way it's easier. At the end, I just want to review quiz 1 to 4. I just want to say that everything on the slide is part of the quiz. If it is not on the slide, you don't need to know those, even though those are on the reference book or textbook that I refer to. Because when I set question, I just look at the slides. I don't look at the things that you probably need to read for your understanding in the textbook. So I would suggest that when you prepare for your quiz, you should just go slide by slides and try to understand. If there is anything that you can't understand and it's written there, the textbook will help you understand that. So I'll give you some I may give you some practice quiz questions on mass balance. Actually, I'll give you, which is just for your practice. I'm not grading them, but the quiz that will be on Wednesday after the class that I release is 10 questions, 20 minutes. So usually it's supposed to be 12 minutes in the last when there is a in-class exams, but I give you 20 minutes. If any of you need more accommodations, I receive your emails or through the Center for Accessibility, and you will get that. I realize that CCLE is not didn't give me options where I can increase the time for specific students of need. For that, what I'll do is I'll use everything will be on campus wire, because it allow me specific time. So I'll release the quiz on campus, not campusware. I'm sorry, Gradescope, um, Gradescope or campusware. I'll check that one. Okay, I think Gradescope, but Gradescope is linked to the LinkedIn, not LinkedIn, the CCLE. So as long as you have CCLE, you just go there and Gradescope will be activated, like it is activated for the um, for the um, for the homework number 2. You just have to do the same thing. So again, it's a 24-hour window. So that means if I release whatever time I release, you have 24 hours to appear in the exam. But just know that you have 20 minutes that you need to take is part of 24 hours. So that means if you take exams just 10 minutes before the 24 hours window is closing, you'll not have enough time. Um, so those are just the basic thing.
Hey,
Yeah, Joe, you're
there's a career fair. There's a career fair on Wednesday. It's gonna be a pretty long day for me, and I'm sure a lot of other people in this class. I understand it's 24 hours. Do you know when you're releasing that? Is it the start of like when class would be?
so let's put this way. I'll release that at 5 o'clock on Wednesday to make it easier for you guys, because then you don't have taking any class, and then it'll be due next day 5 p.m. So that will be Wednesday 5 p.m. to Thursday 5 p.m. So that way you are not
Thank you.
affected by any kind of activities. Um,
Is the quiz going to be multiple choice
it's multiple
or
choice.
is it okay?
It's multiple choice, but that multiple choice has numerical answers. So that means you may need calculators or stuff nearby. And again, if you are planning on um, reading everything during the quiz, you will not have sufficient time, or it might take a little longer. So that's why I say prepare for the quiz beforehand. And anything you need
Okay,
to write,
sir.
write those in a page so that it's accessible to you. Again, that just makes it easier because it's open book. Yes.
Professor, sorry, you said we have to submit something on Gradescope. So the numerical solution, we need to take a picture and upload them on the Gradescope?
No, no, no.
No, no? Okay.
You can solve those separately, but just making that choice A, B, C, that's a numerical answer. So that means you need that calculation somewhere in your place. I don't need to see that, as long as you a correct answer will receive full points.
The quiz is gonna cover lecture 1 to 5 or just 1 to 4?
1 to 4. So today lecture is not part of those things. So anything that
Okay,
cover in the last 2 weeks will be part of quiz.
okay, thanks.
Okay. All right. So if you don't have any questions, we'll get to the today's class. All right. So application of biology. Um, Let me get to the different point. So when you think of environmental engineering application, in fact, environmental engineering is born because you want to treat our wastewater. And that's the reason environmental engineering was the discipline developed in 1970s. Um, so wastewater treatment plants at the core is a biological treatment. So secondary treatment system is a biological treatment system where you aerate with oxygen or air, and that oxygen is it's like we breathe. Same thing, microorganism will take that oxygen, breathe them in, and start digesting the food, which is the chemicals for them, all the organics, oils and grease, all that stuff that's there, organic stuff. So that's how they degrade it, and that's the wastewater treatment plant. And then same thing, if you think of drinking water treatment plants, a lot of times people use simple sand filter. So in that sand filters, where you just pour water on the top, over time the biofilm will develop on the surface of sand, and that biofilm can actually remove a lot of chemical pollutants and degrade them. So that's why drinking water treatments is also related to some context biological treatments in it. In fact, if you think of in these two systems, we are using biology as a helpful way to remove. They are our friends. You are using them to remove chemical pollutants. But in some cases, those some kind of microorganism can be also very pathogenic or deadly to us. So in that context, biology is a danger for us. You remove viruses, for instance, coronavirus. They are not helpful for us, so you have to eliminate them. So you have to understand also how to degrade or remove those pollutants. So those are disinfections. Disinfection is related to indirectly related to biology, because there you find UV lights or you add some chemicals to kill those bacteria or pathogens or viruses. For instance, people say that for coronavirus, spray your hand, wipe out with a soap. Why soap works better way than even any methane, any alcohol. You don't even need alcohol. If you just wash with soap, it can kill many viruses, or particularly this kind of viruses. So those are things you'll learn if you know the how virus is made up of. Then the other bigger component of biology is the application is remediations. Remediations of soil, contaminated soil. It works the very same way. The difference is it is in soil as opposed to in water. Then also you have to think of infectious disease transmissions by air and water, which is a perfect example is coronavirus. So here is the one application, transmission of infectious disease. For instance, this is particular disease. The case occur in Disneyland here in California. So there was 22 Legionnaire cases. There is a particular disease caused by the Legionnaire bacteria. So they found that it's maybe originated from Disneyland. And the reason they said Disneyland is not Disneyland is just the fact that Disneyland requires lots of cooling, so they have a big cooling tower systems, and those cooling tower system a lot of times harbor or breeding ground for this kind of bacteria. So now question is why these cooling towers are a perfect place for bacteria to grow. Then you have to think of what bacteria need to grow. They need very good conditions, temperatures. So warmer temperatures are helpful for them. And if you think of what is cooling tower, it does bring all the hot water and try to evaporate them, spray them. During the spraying, that evaporations make them cooler, cause them to cool down. And so that's how they maintain the warm temperature or lower the temperatures. And but during that mixing time, you see this warm temperature is there. So that warm temperature is very good for this particular bacteria to grow. And then also cooling tower systems has a lot of nutrients. If you don't disinfect this bacteria or remove those nutrients, sometimes they build up, and that's how you can have those bacteria grow. So that's why it's very important to know how to disinfect them, maintain them. So that's why this is just one of the reason. So as an environmental engineer, you got to understand how bacteria grow, where they can cause disinfection. This is more recent examples. Tracking COVID-19 outbreak by monitoring your wastewater treatment plant, wastewater. So now think of the biggest challenge. How do you know that there is an outbreak that going to happen in this dormitory? A lot of times the outbreak only when you already have infections. For instance, most students or most residents are now get tested positive for COVID. Then only you know that there is infections. But if you know how virus cycle work, before even the person knows it, that's growing, the virus is growing in their body. They didn't even know that. So how to catch that before that? So when our body is creating these viruses, the viruses are growing inside our body. They are also everywhere in our cough, in our body, or in our saliva, in our most importantly in our poop. Okay, so that means even though we don't know that we are infected, we are releasing them through our poop in our sweats or in our restroom. So now if you collect those sweats and you see typical concentrations, if a lot of students are now infected, their poop will have very high concentrations. So you can see that over time, if you're monitoring, they are increasing. Before even the people know it, that they're infected, you see that curve, increasing curve. So by just monitoring the wastewater that are released from the system, we'll be able to identify a potential outbreak before even you find that in normal method. So that's why you see this is a very good application of environmental engineering, where you just take a little bit of wastewater and then look for that the DNA or RNA, amplify them, and then you see you find those concentrations. By far the biggest applications is wastewater treatment plants, where as you can see most of the organic comes here, and those organics or dissolved organics goes to this aeration tank where you just bubble through it, and that air is basically oxygen for us. We need oxygen to breathe. That's exactly same thing for this aerobic bacteria. They need oxygen. They breathe in oxygen and destroy that or decompose that uh, organics, which is oil, all stuffs of organic. And so once they destroy that organic, then you carbon dioxide will emit, and then rest of them will have lots of microorganisms because they are growing. So there we have lots of bacteria here, and so you settle them out, and then you recycle some of the microorganism back because you need them to continue degrade, and then rest of them you can put them as a sludge. So as you can see, this is the way it works is basically biological system. In some other cases, you don't need to aerate them because aeration is very expensive. In fact, 90% of the cost for any wastewater treatment plant is just this pump aerating, because that's the high energy intensive cost. So a lot of times you don't need that now. What they do is they say, okay, why do we even go for that conditions? We can actually make it anaerobic, means you don't need aerations. You just need methanogens bacteria to actually produce methane. And the idea here is that they can take carbon dioxide, or they can use different system to act as electron acceptors or donors. So the advantage here is that you don't need oxygen, and you can also produce methane. Instead of CO2 as a byproduct, you are producing methane. So that means you can actually have energy from the biogas, or this just methane. That's a net energy. Instead of having this as an energy sink, that means you need energy to maintain this wastewater treatment plant. In this case, you don't need energy. Another advantage is that because it's anaerobic, they're not so efficient, they're not growing that much. That means you are not producing so much sludge. Sludge means the things that leave the system here, because those are waste. You have to deal with those waste. Here you don't have to deal with waste. But there are advantages, disadvantages. Anaerobic systems are not very efficient. They are slow. But over time, we learn about them. So things are getting better. So now many of the wastewater treatment plants employ this combination of both. That way they can produce the energy, and we use those energy in the wastewater treatment plants. So many of the wastewater treatment plants are actually a energy producer, not a energy sink. And the last one, or not last one, biodegradation of plastic. This is very important innovations in this new, the plastic is a big pollutant. The plastic is also organic. The organic that you destroy in wastewater treatment plants are not plastic. They are just basically our poop, a lot of things that's easily biodegradable. But plastic are not. That's why plastics are everywhere and they are not being degraded. So now question is, how do you degrade them, or how do you make plastic that is biodegradable? So these are the research you will understand if you understand microorganism, as well as the environmental engineering aspect of the degradations. And then the by far 30% or 40% of the applications is bioremediations of contaminated soil. It's the same principle. You need a substrate, which is food for microorganism, which a lot of times you have to provide as a sugar, methanol, all that stuff. If they don't have enough food, the idea here is that when you let the microorganism grow, and while they are growing, they will produce enzyme, and that will destroy the pollutant. So as you see, these are all different applications, and those applications are very essential for designing different engineering systems. So that means you've got to understand how a microorganism works, functions, very basic level, not very in depth. But if you are using that as your career, you got to understand more complex um, internal, like how proteins and what kind of proteins, all that stuff. Any questions so far? All right, so let's have example just to see how you can apply this principle to solve these questions. Again, this is the questions and this is the answers. Okay, this is examples in your textbook, or not textbook, the reference book that I shared. So it says that 1,1-DCA, that's a particular chlorinated solvent, chlorinated hydrocarbons. So chlorinated means, hydrocarbons means just there are a lot of carbons, usually hydrogen, for instance, methane. But in case there is a chlorine replacing that hydrogen, that's called chlorinated hydrocarbons. Okay. So DCA, dichloroethane, basically there are two chlorine. I'm sorry, let's see, two chlorine. Ethane. So ethane. So that will be another hydrogen. Okay. So the point is, there are chlorinated hydrocarbons which are used for solvent or many different applications, and they are extremely persistent in environment, and they are pollutant, they are carcinogenic. So that means now that it's there in the world, how do you destroy them? So this is what is written here. It is sparingly soluble in water, but miscible with organic solvent. because organic is not easily soluble in water. It is used extensively in chemical manufacturing as a solvent, degreasing agent, as a fumigant in insecticide spray, and fire extinguishers. It is regulated both by the US EPA as a drinking water contaminants and OSHA as an outdoor air pollutants. Because they are volatile, so that's an air pollutants because they will be in the air. You're working as an environmental engineers and are designing a groundwater remediations for a site contaminated with 1,1-DCA. You wish to determine if 1,1-DCA has the potential to be degraded microbially or microbiologically by sulfate-reducing bacteria. How do you accomplish this? Okay, so that's a long question. The point here is that you have a location which is polluted with a DCA and you have a solution. You are saying that I can degrade it if I have bacteria which does sulfate reductions. Means sulfate reduction means if you think of sulfate reduction, sulfate reducing bacteria. So that means sulfate will receive electrons and it becomes something else. For instance, here is hydrogen sulfide. Um, so or other way, hydrogen sulfide becomes sulfate. So now why it is important? If you think of how people degrade dichloroethane or how bacteria degrade, it's a reduction process. Reduction process means they receive electrons. So that reaction is given here. Okay. If you see, that's the um, 1,4 uh, 1,1-DCA. That's the chemical structure. 4 hydrogens, 2 carbon, 2 chlorine. And if you balance that reactions, what it does, it remove 1 chlorine. Okay, by by reacting with the protons or hydrogens, and it receive 1 electrons. So that means, what does that mean? It means that it need electrons to be degrade or remove that, kick out 1 of the chlorine from the that contaminants. Again, these hydrocarbons is toxic because of that chlorine. So your goal is to kick out that chlorine. So what it does, it replaces that chlorine with hydrogen, and in that process, they need electrons. That's why it's a reduction process. So now you know a couple of things. This reaction, for that to happen, you need to have electrons and need to have this one, protons. So these electrons, somebody has to give that electrons. So you need a process that release electrons. So you need a reaction that can give that electrons away. So that's why if you see um, in these reactions, if you have hydrogen sulfide and water, it becomes sulfate and it releases 1 electrons. And that electrons can be consumed here in the other reactions. So that's why it says sulfate reducing bacteria. So this is the bacteria that does these reactions. You could use them. Now, second thing is you also need, if you see this another extra addition here, H plus, this reaction occurs a lot now if you have lots of protons. So that means you have to supply that protons. It turned out this particular bacteria can also, this reaction also release protons or make it acidic. So now your goal is to see whether this is feasible or not. So this reaction will be usually given. So those reactions are given, and this delta G is the free energy. Free energy, the more the free energy, the easier it is for that work to happen. Okay. So the negative free energy means it can occur spontaneously or by thermodynamically is favorable. If it is positive free energy, it's not going to occur. So first of all, you see that's a negative free energy. That means it can occur, but you need that much energy. But if you see this one is less free energy. So that means, if you see based on this half reaction, delta G for this reaction would be, if you combine them together, it will be 49 plus 21, it will be around minus 70. Okay. The reaction is thermodynamically feasible because negative free energy. Okay, that's the reason it says thermodynamically feasible. So now where do you get this energy? So that's another aspect. How do you supply those foods and all that stuff to make this reaction feasible. So again, when you write those reactions, you just look at the delta Z. If the delta Z is less than 0, it is favorable. That's all you are calculating here. Okay. Um, and second thing to also understand that just because it's thermodynamically favorable doesn't mean that it will occur, because sometimes you need that activation energy, which you learn in later of this class. For that, you have to supply some amount of enzyme or activations by either increasing temperatures or some other form. All right, so any questions? These are all for application gone. Now we are going to just briefly go through the type of biological organism.
Oh, about the free energy. Did you
Yes.
say that as it increases, it is I didn't hear what you said after that.
So,
Like what
so
happens when it increases?
if the free energy increases, no. So here, only thing I have to think of is first of all is a sign of the free energy. If it is positive, it's not thermodynamically feasible. Okay, because you need to, you can't do it without supplying energy. Okay. Uh, for that to happen, so anything is free energy is positive, you have to provide it. Nature, you can provide it most of the time, so it may not occur. And second thing is what's the magnitude of the free energy? For here is a minus 70 degree kilojoule per electrons equivalents. So that much energy is required for that reaction to happen. So where that energy will come from? Obviously microorganism has to spend that much energy, and so the ATP, all that stuffs will be used for that. So you have to think of, does the microorganism will get that much energy? Is this sufficient for them to do it? Then if yes, then that will occur. So those are things we'll learn a little bit later. So just for now, just think that no, negative means it's favorable.
Okay.
Okay. All right. So type of biological organisms. Again, this is
Wait,
just
sorry,
very,
Professor. By
yes.
favorable, do you mean exothermic or like
No,
spontaneous?
it means that it can occur in nature.
Okay, so like it's spontaneous. Like
Spontaneous. Yes.
okay, thank you.
How
That
do
means
you
you
calculate?
don't have to modify or apply energy for that to occur. Okay.
How do you calculate?
I think I didn't hear there was some internet lag for me. Can you repeat the question?
I was just wondering, how do you calculate delta G? I don't know if you went over it, but I
Oh,
did like a
okay.
little bit.
Yes, Imam, that's a good question. We didn't really go over through that. So delta G0 is kind of given. Okay. Um, but a lot of times you can calculate based on the reactions, and usually you'll these are things given to you. Um, but there are ways to calculate, but that's not the scope of this class.
Thank you.
Okay. All right, so type of biological organism. This is just a very brief overview of everything biologically possible. You see, we have bacteria, we have archaea. That's very ancient, and I'll talk a little bit about it. As you grow up, it becomes simpler. Bacteria is the simplest organism, and then as you go higher in the tree, you will see more complex. So, for instance, protist, which will be a protozoa, or the oocyst, which has a different cell wall. It has a nucleus, and then talk about plants, fungi, animals, and then humans. Okay. So what we are dealing with here is mostly environmental engineering deal with these kind of things. Okay, majority of this. We use fungi and plants to actually degrade contaminants. So this is more like using them as a way to remove pollutants. Protists or these kind of oocysts, these are usually pathogens. Okay, and whereas bacteria, there can be pathogens, and they may not be pathogens. For instance, you can use bacteria for to degrade pollutants, but some bacteria could be toxic, like Legionella disease, that caused by that particular type of bacteria. One thing you notice here, I didn't write anywhere as virus. Virus is not an organism. It's a dead. It's a it's a vector. Okay, so a lot of times, virus is created as a mode of transmission of genetic materials. So it's not live. It's basically an enclosure. Think of a baseball, or think of a uh, football. But instead of air inside it, you have these genetic materials, RNA or RNA, that goes that can be released and transmit to another bacteria or infect them. That's viruses are part of the evolutions, because a lot of times you cannot transmit that genetic materials without you know without really having this upstream. But virus give that advantage where you can actually pass on to another without really having your offspring. So that's how genetic evolution happens a lot. The virus is a defense mechanism for a lot of times, but sometimes it goes wrong, like this kind of coronavirus. So that's why virus is not part of it, but we'll cover that a little bit. So again, read the textbook so that you have some understanding. I don't expect you to remember or memorize any of those things. Just know the difference between bacteria, viruses, fungi, all that. Okay. So bacteria, I just wrote here a couple of things. What's important of bacteria for us, environmental engineer. When you think of bacteria, you have to think of a couple of things. What kind of membrane it has, because that membrane will determine what kind of disinfectants you will use to kill them when you need to. And another way, membrane types is a defense mechanism for them. So that means you also have to understand, can the bacteria survive in the engineering conditions that you are designing for them to work for you. So that's why membrane types, gram positive versus gram negative. The idea of gram positive negative is a simple thing. It's a scientist named Gram. He developed a particular types of ink. When they apply that ink, if it color the bacteria or the surface, is gram positive. If the ink doesn't stick with the wall, it is gram negative. It's just a simple way to identify them. But really, what that means is the gram positive means it has a the glycolipid layers. That's the membrane. It's a simple membrane. Okay, that particular types of chemicals or the crosslink is there, and it's very thick. The reason it is thick is because it's designed to protect it, so that means it can survive in very extreme conditions. Whereas gram negative, it has a combination of that as well as multiple functions. So it is more complex. Okay, and the reason is that complexity allow them to move stuffs in and out and many different ways. That, so it's more a little bit complex, but though all are present in nature. So again, for environmental engineering context, you just have to think of those aspects. What is made of, how it functions, what condition it can survive, and why. Okay. So when you read that chapter, just text, just think of that a little bit more. But again, I'm not expecting you to memorize any of this. This will not be part of quiz. But this is important for you to understand the designing of engineering system later in the class. Protozoa compared to bacteria is more, if you see, the cell, and it has a flagella, it has a nucleus, it has a wall that can survive in a very extreme freezing conditions. Or so that's why it's it's deadly. For instance, if you have a oocyst outbreak, for instance, many times oocyst outbreak happen in groundwater conditions. That's usually in Wisconsin, all those areas, because they use dairy. There are lots of dairy wastewater, or the dairy cows and pigs and all that. So they are infected by oocyst, and because of their infection, and those water, contaminated water, goes through groundwater, and that's how the groundwater well is contaminated. So the reason why it is very dangerous is because its cell wall is so thick and so strong that normally they are not they don't die, and they can survive for a long period of time before they can become active again. So that's again just to show that the idea. This is the This is the particular type of one which caused this outbreak in many places. Another is cryptosporidium. That's basically oocysts. That's spherical. That's you see on the D here. And they survive. Algae, something you know. This is kind of bacteria, but it's the difference is the bacteria are bacteria. They don't really they get energy by consuming food, but here this is the type of bacteria they actually take sunlight and do photosynthesis like a plant. So those are all the things are there, but again, I just don't want you to go detail, just know the differences. Fungi, on the other hand, are, think of mushroom. Mushrooms are fungi, fungus that grows in a moist environment. And they are complex organism. They can't, most of the time, they can't get the organic, they can't get organics from many places. So they usually infect other organism to get that organics. Now, that's why if you have a food or something in a moist environment in refrigerators, stay for longer time, fungus will grow because it has moisture, it has organic, so fungi can actually survive there. And fungi is also important because they, a lot of times fungi help plants to supply them nutrients. So those are mycorrhizal fungi, means they survive in the roots. So these are different things we'll learn at different part of this class. Just know that they are different from bacteria. The last one, which is again not microorganism, but I just added it so that it's important for you to understand this whole COVID aspect. So now you see that, for instance, couple of questions like here: why is N95 mask more effective than traditional surgical mask? These are the questions you keep hearing a lot. So let's have the only thing you should remember is viruses are extremely small compared to bacteria or other things that we talk about. The maximum size of viruses is around 200, I would say 200, around that 200 nanometers. Okay. If you think of bacteria as a size comparison, bacteria size is, I would say 2 to 5 micrometers. So that's 1 micrometers is 1000 nanometers. So roughly 1000 times smaller than, yes, around 1000 times smaller than, 1000 or 10 times smaller than bacteria. Okay, um, but virus can be much smaller. Okay, as I said, they have an envelope here, and that envelope is a protein layer, and the inside that you have RNA. That RNA is the genetic materials that makes it infectious. So viruses can have different types of viruses. It's spherical, you can see over here. It's just basically a delivery system for genetic materials. So that's the concept, But now let's think of answering these questions. Why is N95 mask more effective than traditional surgical mask? Any students, volunteers?
Because of
Has to do with the filtration process.
Yes, Ramiro. That's based on the um, size of the filters that N95 has. If you think of surgical mask, that surgical mask, the pore size are bigger than the virus size. Okay, that surgical mask is usually designed to prevent blood or the big big liquids or bacteria, larger particles, not to filters, not the viruses. So that's why viruses can be actually transmitted inside. But now, if this is the case, why people say that why not use N95? Why you are using any mask? Well, if you think of why we are using any mask, is not to protect us. It's about protecting other people, the doses of viruses, because every time the virus transmit is mostly by when we are talking, we are releasing those droplets. That droplets from our lungs, those are the place where virus infect, they get out of the systems. And those droplets can migrate much further now. So those droplet size is much bigger. Okay, that's 2 micron, 10 micron. So if you have something, any kind of cloth, that droplets would be blocked by those masks. That means it will not spread around environment. So it's not about protecting you as much as protecting other. Okay. So if everybody use mask, that means they are not spreading. They are not super spreading those viruses all over. That means you will be protected. So that's one of the reason why people recommend the CDC recommend using mask. Unfortunately, it's a pure science that everybody knew for a long time, but it unfortunately become political, and that's why it's becoming more infectious than before. The second question is how far virus or droplet can migrate in air. Any That's basically 6 feet distance. Wow, they come up with that number. So any students to volunteer to say what controls the transmission or the migrations or distance travel. So think of this is our mouth. Okay, I'm just going to okay. This is really bad drawing, but let's say I cough. My question is how far it can migrate. So what controls that?
If there's any good airflow in the room,
Uh,
or
let's not think of airflow yet, but think of I just cough right now. Okay.
The
So,
particle size?
yes, number one, particle size. Particle size. Okay.
The velocity.
Yes, the velocity of droplet. Okay. These are the two most important factors. You learn that in the later class. Air quality transmission in like a week 9 or 7 or 8. I forgot. Um, think of things move because there is a inertia. There is a force behind it, and that force is a when you do that one, that's the one which apply that force. It's like pressurizing them and releasing them, and that velocity that it attend here, unless something stop it, it's going to migrate long distance before the air resistance itself slow it down. And so roughly when we speak, we don't exert that much velocity. uh, when we speak, it goes to around, let's say, 6 feet. Okay, 6 feet or around a little bit more. If we cough them, it goes even farther. If we when we sing, we have more pressure. We use more energy, so that means it has more velocity. So speaking, singing, coughing, they're all release those viruses at different velocity. um, so that's why if somebody is speaking for a long time, even though the velocity is not higher, they're emitting a lot more because the dose is very lot. Whereas coughing, like a lot goes then stop. So you have to think of all that context. So that means you have to also think of concentrations. But either way, the distance it migrate is based on the particle size. Okay. So I'm going to say the distance. So this distance migrate, I would say the is inversely proportional to the particle size. Okay, because the larger the size, the bigger the weight, the resistance will be higher, and also the weight of itself will make it fall down. So most of the droplets will not migrate more than 6 feet um, on its own without the velocity. Again, that depends on the velocity. That velocity is itself, whether you're coughing or singing or all that. And so now, why mask is useful? Well, mask is useful to retard that velocity very quickly, because when you cough, there is a block here. That droplet itself size is bigger than virus size. So that means the size of the mask, the pore size is usually smaller than the size of the droplet. So that means most of the droplet will be captured. But if you are not wearing the mask properly, that it can emit from the side. Okay. But even though emit from side, it's not the same as speed going this way. It just hit the wall and then it passed through the wall like here. So that means if the velocity has decreased significantly. So that means if somebody is standing even 3 feet from away from you with the mask on, you may not get that exposure because of that retardations. So these are the factors. You'll learn that in this class in later in airflow, but just know the science behind the preventive measure that CDC recommends And I hope that all of you are following it. And now that you learn a little bit, maybe some people who do not understand for whatever reasons or think it's politics of using mask, you can help them understand the concept. All right, so that's all for the applications or the different types of microorganisms. We'll go to the chemical composition very quickly. Any question before that?
Can you just go on the other page for a minute? Thank you. Thank you.
All right, so now let's think of, we talk about bacteria, viruses, all that, but at the end of the day, they are made of some chemical building block. Okay, that's what made our skin, that's our made our cells, or different things. Okay, so those chemistry is important for us to understand why it is important. For instance, if somebody says why we gain weight if we drink a lot of sugar. Okay, so or why we get lots of energy, the high energy drinks has a lot of sugar, what's the reason behind it. So those are the things, that chemistry is related to that. So that's why it's important to know at least the fundamental block of that different part. So when you think of different biological materials, it is made of different types of chemical um, chemicals, and those are, there is some similarity between them. So I'll just go over a few of them here. Carbohydrates, carbohydrates, when I say carbohydrate, basically carbon and hydrogen. So for example, sugar is a carbohydrate. So when you drink sugar, that just basically it's easy to break it down. So microorganism has the easiest way to break it down and get the most energy by when you break it down carbohydrates. So that's why, when you drink the sugar drinks, that gives you energy, instant energy, because your body were able to break it down. And why it is easy to break it down is because of this particular functional group. Carbohydrate has this OH functional group. Those are easier to break than the other one. Okay. Um, and now there are many types of carbohydrates. They are again produced by photosynthesis. That's why lots of fruits taste sweet. So most of the plants materials are made of carbohydrates. Um, Again, there is nothing else to learn here other than the fact that they are sugar, they are easy to break. So anytime environmental engineering applications, if you need to have energy, if you need to give sufficient energy to bacteria or the organism for them to degrade, then give them carbohydrates. But so that means a lot of times people dump sugar in that treatment systems. Okay, if you can get carbohydrate, get methanol or alcohol, not too much alcohol. There are certain alcohol as dangerous, they're based on percentage, but these are the reason you wanted to use. The second type of chemical composition is nucleic acid or RNA, DNA. These are basically the information center. They store the hereditary informations. Uh, for example, the bacteria, the viruses that we just learned, they transmit RNA. Okay, that RNA can also disrupt those, in our systems. So that's exactly the virus transmit that RNA. So the DNA, RNA, they have very complex structures. As you see over here, it has multiple different block arranged different way. That's written here. Okay. And the important thing I wanted to mention, the difference between carbohydrates and nucleic acid is nitrogen. Okay. So you see, this is the nitrogen. This is nitrogen. This is nitrogen. So NH2, NH. These are the functional group that's useful for this kind of nucleic acid. If you see carbohydrates, you never see any nitrogen there. Okay, that's why now if you see what's the main composition of our system is carbon, nitrogen, and oxygen and hydrogen. And so nitrogen is the central at least the most dominant component in nucleic acid, same as proteins. Proteins are basically amino acids. So when you say amino acid, that's basically I say NH2, but there is a this is regular acid. Okay, this is carboxylic acid. Carbox. And this is amino acid. There is an amino, this is amine group. Okay. So that means if you have this acid amine group, so that's in the protein. And proteins are basically the cellular functions. Anytime you have to think of any reaction that for that to happen in our body, it requires proteins. Whether it's a storage, signaling, name it. In fact, most of the all the biodegradations or things that you do in environmental engineering is derived from enzyme. Okay, so this is the by far the most important reason for any environmental engineering applications. So anytime you have to think of what kind of degradation to happen, you have to identify that enzymes, because only when you identify the enzymes, you can actually increase that reactions. So again, the chemical building block is amino acid. So amino and this acid functional group. Um, any questions? There are many other complexities chemistry that I didn't go through because it's not needed right now. But just know that three component: carbohydrates, nucleic acid, which is basically in the nucleus, RNA, DNA, and then you have proteins. Protein is the main reason that you. So this is mostly protein is. Think of enzymes. Okay. This is for reaction to occur. Okay. This is the reaction. This is mostly informations. Okay. So you can take a little bit soil and what kind of bacteria or fungi or all the stuff live in that soil or the wastewater. That way whether your system is having those helpful microorganisms or not. Carbohydrate is mostly we call substrate or food. Okay. So if you want to make sure that microorganisms grow in your system, you have to give some kind of food, and that's carbohydrate. So if you think of chemical engineering, the environmental engineering for biological applications, or biology for environmental engineering applications, you have food and you have microorganism. Then you have energy. Product could be CO2, methane, or it could be anything, biomass, and also microorganism. Because they grow. Okay. And then microorganism produce enzyme. Okay. So this is amino acid. Okay. And what kind of microorganism present is mostly nucleic acid. For instance, if you know whether coronavirus is there in the wastewater or not, you use nucleic acid as a marker. And food is basically carbohydrate. Okay. So that's the basic function. This is why we need to know those three things more importantly than anything else. And one of the things that fun fact is why certain carbohydrates are easier to degrade than other. For instance, another way to think is glycemic index, or another way to say is why brown rice is healthier than the white rice. That's all about this chemistry. Okay. It's based on the location of that in the brown rice, that the coating has lots of that's difficult to digest. That's not an easy sugar. Okay, because of the functional group is very difficult to access. So that's why if you think of plain sugar, it's very easy to dissolve, it's easy to degrade. Whereas if you take those kind of things, those are harder to because they are more complex. So all the chemical formula that you see in this particular slide is not they are all easy to degrade. Okay, I didn't put any chemical formula for the brown rice or that coatings. Any question? All right, so the last part is metabolism, means how bacteria consume that substrate to degrade pollutants, or how they use enzyme to degrade it. So there are two types of metabolism, catabolisms and anabolisms. So that's based on whether you are whether you are growing or you are not growing. Okay, a lot of times anabolism means they produce they take organic compounds and they don't need to degrade it, but they have to because they produce these other things byproducts that's needed for them. So that means those are more expensive for them to do, and that's why they are not growing. Okay, so that's why I say not growing supporting organism. Whereas catabolism, where they actually eat them as a food, so that means they will grow much easily. So those, if your contaminant, organic contaminant, is actually used as a food, that means it's more likely to be degraded in the environment. But many times it's not the case. You apply certain other thing like methanol or sugar in the system, and you hope that these bacteria grow and they produce the enzyme, and that enzyme is not very selective. That means that enzyme accidentally can degrade some other contaminants that present there. So you got to know whether your contaminant is supporting the growth or not. Those are things that makes it easier to understand. And a lot of times they produce, for this reaction to happen, again, these are all redox process. You need a source of electrons. So that could be organic carbons, contaminants, oxygen, sulfur, all those things that we talk about. You could have acceptor of electrons. As I said, no, I'm sorry, oxygen is an acceptor of electrons, nitrate. Um, and electron transfer, for the reaction to happen, you need, again, there are always activation energy that's required, and that enzyme will lower the activation energy. So that enzymes are, we just talk about, that nucleic acid, the the amino acids. A lot of amino acids are enzymes. What it does, it bind with the sites and lower the activation energy for a reaction to occur. So this is all about activation energy. Means, all I'm saying here is, if without activation energy, this is the amount of energy required. But if you add this enzyme, they lower the activation energy. That means that you don't need that high energetic system for the reaction to occur. So that means your system will be more efficient if you have enzymes. Just to, this is not going to be covered at all a lot. Just know that we keep talking about biodegradations. There are a lot of different names for it now. People say biotransformations, biodegradations, mineralizations, co-metabolism, detoxifications, activations. This all depends on what products it forms. Biotransformations means, one pollutants become another types of chemicals. One chemical transformed to another chemicals by using enzyme. Biodegradations, where the molecule or the chemical itself is transformed, but into a very small, simpler organic structures. So that means we call degradations, because now they are not very similar to the previous one. It degrades to very simpler things so that it can also be consumed or used up. Mineralizations means, organic become inorganic. For instance, carbon, all the carbons become carbon dioxide. Okay, so if you have for instance, if you have a nitrogen amino acid, it become amine or ammonia. Okay, so when that happens, that's become mineralizations. Co-metabolism, all that stuff. Again, you don't need to understand all that details, but just know there is a difference people use at some times. They're all same process. And for this, we'll learn a little bit more in the, I think, next class, or when you talk about waste management. This is something we'll learn a lot, where these are the different types of waste you see: BTEX, polyaromatic hydrocarbons, nickel, zincs, all these metals. How do you fix them? How do you make sure that they are not spread in the environment? So those are the times you will apply this concept. Right now, you don't need to remember. But this is something will come across a lot at that time. Just I want to just highlight here. A lot of chemicals, when you see chemical contaminants, you can, just by looking at the chemical structure, you'll be able to say whether this will persist in environment, whether it's a difficult one to degrade. One of the things you should look for, like I said, some sugars are easy to digest than other. This is the same reason. If you have any kind of halogens, fluorine, chlorine, bromine, iodine, these are the halogens. That's the in the periodic table. If you think of the very last, that's inert gas. The one before is halogens. So if you have any of these halogens, chlorine, fluorine, bromine, replacing hydrogen in the hydrocarbons or the organic pollutants, it become more persistent. Because they are highly electronegative, they makes the bond very strong. That means bacteria or the microorganism need to spend a lot more energy to break that bond. And usually they don't want to do that, because why to spend so much energy in something when they can just eat sugar and get that energy. For instance, if I say just eat something that doesn't you can digest, obviously you will not eat that unless if you're looking for high energy very quickly. So same thing here. No? So if you have lots of these halogen compounds, that will make it persistent. So fluorine is more electronegative than chlorine. That means fluorine carbon bond is more strong. That means more persistent. It's very difficult to remove. Um, Second thing you have to think of is, if you have more halogen, that makes it more and more strong because it stabilizes the bond. Same thing if you have branched. You'll. I'm getting too much into organic chemistry. Will not don't remember any of those things right now, but we'll get to that point when we talk about specific wastes. So these are the basic formula. If you know these things, you'll be able to say which is more biodegradable or which is more persistent. Lower. So let's see one example. If you think of this is organic pollutants, what you see is this is just a carbon. This is a notation if you take organic chemistry. The thing that you take away from this class is you have this chlorine. Okay. And that chlorine makes it very stable. As I said, if very stable means persistent and makes it very difficult to degrade. And it turned out this particular structure was is actually very toxic to mosquitoes. So this person who discovered, that's called DDT, that's the because of discovery, there are a lot of mosquito, the malaria occurrence decreased significantly because millions and millions of people are dying before with malaria. But application of DDT actually saved those millions of people per year. And that's why the person who developed this one, Paul Hermann Muller, he got a Nobel Prize in 1948 for this medicine. But think of this, that time it was all good. But 1960s, when the Rachel Carson, the person who actually kind of accelerated the process of developing environmental engineering as a discipline, she found that because of the DDT applications, lots of these the fish are dying, or they have their genders are being changed, they are mutating. So she discovered that, and because of that, DDT is banned. So what is very good and save life in one place is actually become toxic. So as an environmental engineer, a lot of times what you have to do is look at the chemical structure and try to figure it out whether this can be future toxic. It could be toxic in future. If you heard about PFAS, that's another group of contaminants which has carbon fluoride bond. Okay. So those are the things used for extinguishing fire in a petroleum fire, and they are also used as a coating in your cookware. For instance, Teflon. Teflon is a PFAS, and so those are very resistant to heat, resistant to all kind of chemicals. That's why I say no. Anytime you have fluorine, it's very strong bond. Now, well, they use for a long time, but like DDT, they have found recently that cause cancer, and they are so persistent that they are found in every groundwater. So again, these are the things that you learn. You got to understand chemistry a little bit so that you could be able to predict those persistent or try to understand. One other example. Let's see. This is example quiz question. But again, I'm not going to ask this quiz for this class. When we go for the actual waste remediation, that's where you'll understand this. But based on the what we learn, uh, let's see how many of you think or for sake of time, let's see if I can make a poll. So look at these questions and see if you which one is the answers among these chemicals, which one will be more persistent. So let me make a poll very quickly. Let's see, edit. I'm just going to say question on slide. Your answer would be A, B, C, D. Okay. Okay, so let's see. Save. Let me launch the poll. So let's see. All right, so let's see what's the answer. So I'm asking which one is more toxic, or the persistent based on this here. Okay. So if you see here, the things are more toxic or the more persistent if you have more fluorine halogens, and if you have more halogen atom for carbons. So as you see over here, halogen is chlorine. You have one chlorine here, you have two chlorine here, you have three chlorine, you have four. So that means this is the more toxic, and 98% people say yes, or 99%. Okay. Right. So let's see. Among these chemicals, which one would be more persistent? Again, look at the number of halogens as well as the type of halogen atom. I'm going to bring that. So the difference between these two are, if you see, carbons are same, hydrogen same. The difference is it has two chlorine, it has two fluorine. Okay, or two fluorine. So now if you see this original, think of the first one, persistent increase with or decrease with if you go to these directions. Fluorine is more electronegative. So that means anything with fluorine will be very much more persistent than anything else. So that's why if you see, the answer is A, because it has fluorine. Oh, I'm sorry, answer is B, because it has fluorine. And 80% students chose this answer. I saw that 7% students chose answer D. There is no D here, so that means maybe you didn't see what the question was. All right, so I think the last thing we're going to learn before we say this is the end of this class is the math behind the growth. As I said, the enzyme is produced, and that enzyme production depends on a lot of factors. That enzyme is also produced by microorganism. So how do you ensure that there are more enzyme produced in a reaction? That's the one that you have to design as an engineering system. So that formula is here. The velocity of the enzyme is the amount of enzyme, or the rate. Think of rate, okay, enzyme action or rate is certain amount, the maximum possible for that type of microorganism, times the substrate. Substrate means the food that are available for them to produce that enzyme, divided by Km is like a half saturation constant. Sometimes different people use. What that means is plus S. That half saturation means this. Now, if if you have more substrate, okay, the growth rate will increase. Okay, that's what the enzyme production rate will increase. But after a point, if you have more and more food, after a point doesn't matter, because there are other things that will limit it. So that's why it will achieve a asymmetrically it will be achieve a maximum. So what it means that how much amount of food is required to increase that uh, increase that action rate or activation rate half of the maximum. So Km is equal to B, or the food, if B equal to B max by 2. Okay. So what it means that how much food is needed for that activation energy to be half of what is maximum. Why it is important? Because if the Km is small, that means you don't have to provide lots of food for that reaction to be efficient. And so you usually want Km to be very small, so that even small amount of food available in the environment, they could actually be more efficient. So that's the reason you use this math. Okay, so you're looking for Km small efficient, it means, and efficient at causing the reactions at low low food environment. Okay, so that's the whole concept. And this is the same thing applied in wastewater treatment plants, where you are looking for the growth rate. So where the growth rate is mu, that's the growth rate. Mu means growth rate. Same thing, if you have lots of food or substrate, the growth rate will increase. After a point, it will become saturated. Okay, so this is mu max. So Ks or Km, a lot of times people write K saturation constant. Ks is the same concept. In this case, in wastewater treatment plant, you want Ks to be high. Okay, the reason is why. Let's think of Ks is high means this. So that means this is Ks1, Ks2. Let me erase this one so that it's less confusion. Ks2, this is substrate. The unit of Ks is same as substrate. So let's say why this is good. The reason is, here you don't want the microorganism to grow a lot, because it grow a lot, what happens is you produce lots of microorganisms as a sludge, and you don't want to deal with those sludge or waste. So you want microorganisms who do not grow a lot or high metabolism, and they destroy all the organics they have. They eat all those. So that means, think of another way. Some of us are very lucky who can eat a lot. I'm not that. I can eat. I can say that I eat little bit amount. I can gain a lot of weight. So that's, in that case, my Ks is low. Okay, my metabolism is very low. So that means I can gain weight much quicker, even small amount of weight food. So that's something you don't need in wastewater treatment plants. You need microorganisms which will not produce as much, not multiply as much, or not grow as much, but still they do the work for you. That way you produce less waste. So Ks high is favorable in wastewater treatment plant. Okay, WWWTP is wastewater treatment plant. Okay, so that's WWTP. All right, so that's all for this class, and there are this example I just want to mention that is on the class in the is in your textbook or the reference book I gave you. So those solutions are given. That's using the concept you learn in this class. So what I'll do is Any question about this class, about this class before I stop the recording and restart another for just for the sake of quiz?
Professor,
Yes.
would you mind explaining the chlorine and fluorine situation again? The previous quiz question.
Okay. All right. So the what is more persistent. Okay. So I didn't explain a lot, so that's why it's difficult to understand conceptually. But if you see what this slide means, it says if you have lots of hydrogen, so hydrocarbons, that's all you're talking about, carbon, hydrogen, okay? Those are easy to degrade. But when you replace carbon, hydrogen with another halogen, let's say chlorine, these are things can happen. That makes the bond much more stronger or very difficult to degrade. The reason is when you think of carbon hydrogen, what's the difference between electronegative? Because when you think of when the bond can be broken, it's based on how strongly they held onto each other. So think of fluorine. If you remember the very first class, electronegative. Fluorine is the most electronegative atoms in the periodic table. Okay, in the
Oh,
periodic
I see.
table, if you go left to right, electronegativity increases. Bottom to top, electronegativity increases. Fluorine is at the top right. Okay, so that's why it's very electronegative. It's very electronegative means it has very high affinity to electrons. So that means the fair electron is strongly bound towards fluorine as like really strongest bond. So it's very difficult to degrade it because of that bond. Okay. Um, Next best is chlorine. That's why anytime you have chlorine and fluorine, they are persistent. And that's the reason all these chlorinated solvents are a problem for environment. As you go bigger and bigger, bromine that's become less and less. And the question why fluorine is more electronegative is because if you think of fluorine, it has seven outer shell electrons, and you just need one electron. So that means you have very strong affinity to have that one electron. On top of that, it's a very small atom. Because a small atom, nucleus is very close to the outer cell. So that's why they attract very strongly. So they will not let it go. Whereas chlorine is bigger, bigger. So that's how it goes. But again, all you have to think of here is either fluorine or chlorine. Okay, fluorine is more electronegative. That means that bond is more stronger, persistent.
Okay.
Any question?
Yes, I have a question. I'm not sure what is S. Is that a concentration?
Question. Well, let me see. Are you asking about this S?
Yes.
Yes, this is substrate concentration. So that means it could be glucose, it could be methanol, it could be anything. So S stands for substrate or food. Okay. And this is a notation for concentrations. Okay. So anytime you see this bracket, it's a concentration term.
Okay. And what would be the unit of the rate?
So that would be unit of rate.
Yeah.
That's like think of any reactions. Think of what is the reaction, d by dt, right? That's the reaction and the growth rate times. So think of this one. If it is a first order reactions, the unit is 1/time. Okay.
Yes.
If it is first order, so I was just going to write mu1 is the first order. This is 1/time. If it is zero order,
Okay.
this will be concentration over time. Okay.
All right. Thank you.
So that's the same as before. It's not different. Okay. Just the based on the, I didn't tell what order reaction it is here.
Okay. Thank you.
Any other question? All right. So I'm going to stop the recording.